maximum likelihood. So here we have a couple of different trees. These are the different trees that you could make out of these sequences. And based on this, we can say, all right, what's the maximum likelihood? So we could have a tree where we start with T, and we change it to a G, and that carries on to both of these branches. But on one branch, we change to a C. And so basically, what are the odds that a T can change to a G, then stay the same, versus a G to a C? What are the odds of those changes or remaining the same? Versus, what are the odds of a T turning into a G, turning into a C, turning back into a G? What's the likelihood there, or what's the likelihood of T turns into G, which is preserved, or turns back into T, which then turns into C, which then also turns into G? I mean, this one, you've got the most changes happening. And so unless that is a really common change that happens a lot, like a transition rather than a transversion, I would say that's probably the least likely tree of this bunch. Yeah. So in statistical terms, statisticians understand how this tree works relatively well. It has an advantage over parsimony in that you can estimate the evolutionary history and a pattern, and you can say, oh wow, these things evolve faster than the rest of the tree, slower than the rest of the tree, in larger steps than the rest of the tree, something like that. You can take into account probabilities of changes between the two purines versus the purine to pyrimidine, stuff like that, transition, transversion. You can also eliminate the problem of long branch attraction, because you're not just trying to make the shortest tree, you're trying to make the most likely tree. And so one of the differences you see between a neighbor joining and a maximum likelihood tree may be the effects of long branch attraction. You're not going to know for sure that that's what it is, but this is a worthwhile thing to discuss, because this is a thing that has to be in your mind when you're using those simple methods. It's like, oh, am I going to start attracting my long branches together just to get the shortest tree? Yeah. All right. And it's also using sites that would have been uninformative under parsimony, because we're using sites where the G could stay as a G instead of changing. And so that's why this is probably the best tree that we could make out of those. Disadvantages: it's slow, it takes a lot of computing power, it can take you days, weeks, yeah, years, unless you're running on the biggest cluster available. And so that may just not be practical for the type of analysis that you are trying to do. Also, if there's missing data, or if you have lots of data from one thing and very little data from another thing, you may generate trees that don't make a lot of sense, because generally these trees work better with lots of intermediate steps that you can calculate that join everything together. We like these little very branchy trees rather than trees with a lot of close short branches and a few long branches. Those are not preferred. Only questionably applicable if you're dealing with anything other than DNA or protein, because what's the rate at which a nose changes? I don't know. Measure my parents, measure me, measure my kid's nose. I guess we could estimate it, but that's only for our particular genetic background. There may be genetic backgrounds where it can change an enormous amount, overnight, in a generation. So we can't necessarily assume that any other change would actually fit our evolutionary parameters, our sort of model of evolution. And so those may not be useful assumptions. And yeah, it's a little harder to get your head around, so it's maybe philosophically less well established. Whereas like parsimony is like Occam's razor. Like, simplest explanation is the best, bro. You just deal with that. Because you can try it out in other walks of life, and you know what? Usually works. It's actually pretty good advice. But maximum likelihood as calculated based on certain parameters that I came up with using other methods, It's kind of dependent on how good were those parameters, how good were the methods used to generate those parameters, how much data did you have in when determining those, and that's going to have a giant effect on the outcome of your tree. So, back to this, like a little overview before we go Bayesian. UPGMA and neighbor joining are going to be clustering algorithms. We're going to seek to join things together, either from our star, and that's in neighbor joining, or from our little dots on a grid that we're putting circles around to join them together, and that's going to be the UPGMA. They're usually not acceptable for publications anymore, just because science has moved on, if you can believe it, since the 1950s/1980s when these things came out. Maximum parsimony and minimum evolution, these are maybe acceptable for publications, although in general, these are also kind of rough draft methods. If you have a bunch of sequences and you just want a general idea of how they're grouped together to then base other work on, this is fine. It'll get you a quick answer, and it'll be somewhere in the ballpark. Generally, though, also not preferred for publication. Most publications would want you to have either a maximum likelihood method or something like a Bayesian method, or something even more sophisticated. There are more sophisticated things that kind of go on top of these. All right. So, the next chapter is only like 2 slides long, and we'll have a separate thing online that helps explain Bayesian math, because it's pretty neat, and it has its own set of problems, but they're kind of a different set of problems. And in general, this is considered to be a more reliable method to use, particularly when you have nice, thick data sets with lots of data in them. And it's still pretty fast. So, yeah, it's a way of sort of shortcutting the process and calculating your maximum likelihood in a different way using a surprising mathematical trick. So, stay tuned for the next section where we discuss that.